First of all, we can also do single cell analysis using this kind of devices, and we can precisely control the fluid flow. So we can also integrate some analytical techniques into it. Next thing, protection against contamination and evaporation. And we can also do online monitoring. For example, if you're running a reaction, you can do online analysis too. So basically, it's a miniaturized lab. The concept originated from integrated circuit. So similarly, we integrate multiple functionalities in a single chip like this. So it's a traditional laboratory where we do reaction in separate chamber. Then we have to take the sample out for purification, centrifugation. Then later we will send the sample for analysis. For instance, NMR, IR, Raman, everything separately. But in this case, we can do all these functions using a single chip. That means within a chip, we can prepare a sample. In the chip itself, we can purify, and also we can detect the products, or in some cases, we can detect the intermediates also. So let's see the history of microfluidics. Basically, photolithography has been invented around 1950s, but in the beginning, the concept was mainly used for circuits and engineering field. But in 1990s, Professor Andreas Munz introduced the concept microTAS, that means total analysis system. Then in the beginning, we started using glass and silicon-based microfluidic devices for chemistry. Then in 1990s, especially in 1998, Professor Whitesides introduced PDMS for microchip fabrication. So nowadays, we can use polymers like polyethylene terephthalate or polystyrene. We can use those polymers for the fabrication of microfluidic devices. And nowadays, the emerging fields are like paper-based microfluidic devices, organ-on-a-chip technology, where we can make some particular organ inside a chip, and we can nicely control the parameters. So now it's towards human on a chip. So let's see the figure. It's about the number of publications in this particular field, microfluidics, in engineering and in multidisciplinary, and also in chemistry and biology. Here you're seeing a microfluidic chip. It's a coin, you can see the size. So it's basically like a microscope slides, 2 microscope slides. We can make channels on one slide, then we have to bond it. So what are all the functions? So first, we can perform a chemical or biological reaction. In the same chip, we can do separation like chromatography. If you are doing it on a chip, then it's chip chromatography. Then electrophoresis, isotachophoresis, we can perform these.
Excuse me, sir, the slides is not visible, sir. Please enlarge the slides. The presentation slides, sir. It's not visible. Okay, sorry for the disturbance, sir.
Okay, maybe I can stop share and share it again.
Okay, thank you, sir.
Actually, it is visible, no, sir. Brinda?
Visible, right?
Yes, ma'am. For me also it was visible, ma'am.
Yes, sir, actually you need to pin. The person who informed, you need to pin that slide, sir. Then it will be visible to you. Sir, you carry on, sir.
Okay. How about now? Is it visible? Hello?
Sir, now...
Now it's not there, sir.
Not...
How about now?
Yes.
Yes.
Yes, sir.
Okay, fine. So
Okay, sir.
we can.
Okay, thank you.
We can perform these functions like separation and detection. We can integrate Raman spectroscopy fluorescence, IR, and we can also do some electrochemical measurements. And finally, we can send the samples to mass spectrometry. So these are all the possible options. So what are all the materials can be used for the fabrication? So in the beginning, originally researchers used glass and silicon, but the process was a little bit complicated. And later on, in 1990s and in 2000s, researchers started using these elastomers. So basically the preparation procedure will be a little bit simple. So now we can use PDMS, polydimethylsiloxane, or Teflon, PMMA, polyester, or polypropylene for the fabrication. Also, we can use paper, that's paper microfluidics, textile microfluidics. But the user should decide the building material, because for some chemical reactions, these polymers are not suitable. So we have to use glass. Even for separation process like high pressure liquid chromatography, we prefer glass. So based on the application or the experiment, the researcher should decide the building material. So based on material, we can divide the microfluidic devices into 4 categories, like inorganic based, polymer based, paper based microfluidic devices, and textile. On the other side, we can also categorize the microfluidic devices based on the propulsion behavior. That means how the fluids are flowing inside the channel. If the fluid is continuously flowing inside a channel, then we can call that as continuous microfluidics. Other option is droplet microfluidics. We can make a really tiny droplets inside the microfluidic channels. So then it's called droplet microfluidics. In that category, now we are started using digital microfluidics, where we use electrodes to manipulate the fluid. Really tiny droplets like microliter, nanoliter, even picoliter is possible. Another one is centrifugal microfluidics, where we use centrifugal force to manipulate or propel the fluids. So there are a few steps. First, we have to make a design, how our channel should be, and what are all the functions we are going to integrate. So we have to use a software to make a design. Then we may need a clean room facility and also some sophisticated instruments for the fabrication procedure, like lithography, photolithography, and we have to make channels on slides. Then finally, we have to bond 2 slides using high temperature bonding, and there also we got different techniques. And finally, we have to give inlet and outlet to inject the samples or reagents into the chip. Here you are seeing a typical microfluidic chip where they got electrical connections too. So one slide and the top slide, here you got channels like this, and electrodes also. You can integrate electrodes for electrochemical measurements if you are planning to do electrochemical measurements. Then we have to have inlet and outlet like this to inject samples. So I would like to show 2 common procedures to get some more understanding about the fabrication, how to make a chip. So in this case, 2 glass slides. We need 2 microscopic glass slides. So on one slide, we have to coat sputtered, like we have to sputter chromium layer. Then we have to make a mask with proper channel design. Then we have to etch that chromium layer. Then with the help of hydrofluoric acid, we can etch glass and we can make our channels like this. So it will be like our hair. The thickness is usually around 100 to 200 micrometer. Then we have to remove the photoresist and chromium layer, and the other slide, glass slide, should have holes like this for inlet and outlet. Then we have to bond the glass using, in this case, it's a high temperature bonding. Now the chip is ready. We can perform a chemical or biological reaction. And here, a polymer-based microfluidic device, PDMS, polydimethylsiloxane. In this case, we don't need to etch the channel using hydrofluoric acid. So here we have to make a mask, then we have to develop a pattern. Then with this pattern, we have to just pour the PDMS mixture into it like semi-solid condition. Then we have to dry it. Then finally, we got two options. Both slides can be PDMS or hybrid microfluidic device is also possible. Like one slide can be PDMS and other slide can be glass. So we have to decide based on the requirements. I mentioned in the previous slide different types, like here closed chips and also open chips are also possible. In closed chip, that means you got two slides. In open chip, just one slide. In digital microfluidics, sometimes we use just one slide, and with the help of electrodes, we can manipulate the liquid. So in continuous flow, the flow will be without any interruption, just the flow will be continuous. And droplet, we will have two different phases of fluids, and we have to make droplets. In this category itself, we got this digital microfluidics. If we are using electrodes and by applying potential, we can manipulate. And now the emerging one is optoelectrowetting. That means we use laser or light source to manipulate liquids on electrodes. The other side, microarrays, they are also in this category. So this is what I mentioned. So in continuous flow, it will be like this. For example, reagent A and reagent B. So you got two inlets and the flow will be like this. And we have to mix the reagents to carry out a reaction. In this case, it's droplet microfluidics. So we got microreactors like this, a tiny droplet where reagent A and B can react and we can perform chemical reactions inside a microliter droplet. This is what I mentioned. You got two phases here, oil and water. So when you inject two different phases, we can make droplets like this, really tiny, nice droplets. And this can be used for chemical reactions. And also nowadays, scientists are using this for single cell analysis. So a single cell can be trapped inside a droplet and we can detect or monitor a biological process. Applications of this particular droplet microfluidics in different fields, like we can use this for biomolecular assays, digital PCR, cell screening, protein crystallization, and also we can make particles and emulsions using these microfluidic devices. So let's see the other interesting topic, digital microfluidics, where we have electrodes like this. We can perform chemical reactions just using a single droplet. Let's say reactant A, reactant B. We can move this from one electrode to another electrode by applying appropriate potential. And we can mix the droplet inside this electrode, and we can detect, or we have to take the product from the chip. We can do mass spectrometry analysis, or some other optical detection is also possible. The difference between digital microfluidics and continuous flow microfluidics. So in the case of continuous flow, we usually use microliters or nanoliters of sample. In digital, we can go for nanoliters, even picoliters volume. And in continuous flow and droplet microfluidics, we use pumps to push or to propel the fluid. So we have to use syringe to apply pressure, and the fluid will flow through the channels. But in the case of digital microfluidics, we don't need pumps. We can just use electrodes to manipulate the droplets. And here we need bulky test circuits. And of course, in digital microfluidics also, we need circuits. And for the fabrication in continuous flow microfluidics, we need lithographic technique. For digital microfluidics, we can use this printed circuit board, PCB. Applications of digital microfluidics. It's a versatile tool, and nowadays researchers are using this digital microfluidics for biology, chemistry, and also for medicine. So let's look into the applications in different fields like synthetic chemistry, analytical chemistry, and finally biology. So here you can see in the beginning, we use some really big beakers for chemical reactions. And nowadays we use glasswares like this. And we believe now we use microfluidic devices, and future, most of us may use this kind of chip for making or performing reactions. So in this case, automated optimization of reactions and catalysts and solvents is possible. We can program it and they can optimize the reaction automatically. Then even small scale, now we are doing everything in small scale, but maybe in future we can make really big flow reactors in companies also. So you have to just press the buttons to carry out a reaction. And here you are seeing a chip with multiple functionalities like reaction flow reactor, then chip chromatography within 1 centimeter. Then finally, we can send it for mass spectrometry analysis. So why microfluidics and merits and trends? You can check this article to know more about this particular thing. So just an example, now if you are doing multi-step synthesis, like 2 or 3 steps, you have to purify it, then you have to make the next reaction, then you have to do column chromatography to purify it, and you have to take that sample for TLC or NMR. But in the case of microfluidics, you can perform all these functions inside a single chip. So for reaction, first step, then you can purify it using chip, like a chip chromatography. Column can be integrated in the same chip. Then step 2, we can perform like this, and everything can be automated. That is also possible. So these are all the existing challenges and how microfluidics can help with synthetic chemistry. So first thing, manual operation, and we use a really large amount of reagents, waste of time, reagents, labor, in some cases, poor reproducibility, and of course, safety hazards. Like for some chemicals, it's really harmful and dangerous. But when we use microfluidic devices, they are comparatively safe and short residence time. We use minimal reagent, and automation is also possible, and low exposure to hazards. Regulation, when we do manually, the reaction parameters, we cannot control it more precisely, or it's not reproducible. But in the case of microfluidics, first thing, we can precisely control the experimental parameters, and they are more reproducible. Like you can use the same chip for multiple reactions. And integration, that means we have to perform a chemical reaction in a beaker, then we have to take different amount of samples for different analysis like NMR, Raman, IER, XRD, SEM, something like that. But we can integrate at least 4 to 5 analytical techniques on a single chip. Let's say optical detection like Raman, fluorescence. We can do that during a chemical reaction. And the chip itself, we can spray the compound into a mass spectrometer from the chip using different methods. We will see that. So this is from a famous Professor Ensign. And if you are interested in flow chemistry and microreaction technology, you can check this article too. So we can optimize a chemical reaction using a flow reactor or microfluidic chip, and we can also do online analysis. So a picture just showing the possibility of different functions. So if you are using beaker, then we usually use stirrers, magnetic stirrers, to stir the sample, or reagents. But in the case of microfluidics, we have to design our chip like this. So the channels should be like this. If we want to mix it, let's say if we are performing a chemical reaction, then we need to make a design like this, zigzag, or you can choose your own design, but it should mix the fluid efficiently. Then only the product will be good. So designing a microfluidic chip is a critical and important step. Here you can see a picture of automation of chemical analysis with small robotic arms, dawn of unmanned analytical laboratories. That means here we got small robots. They can inject reagents and hazardous chemicals into a reactor. Then we can also perform mass spectrometry. So maybe in future we may have something like this. Even robots can perform hazardous reactions without any safety issues. We can also use microfluidics for nanomaterial synthesis. So in this case, the authors produced nanocrystals. So we can precisely fix the flow, like the rate, and we can make nanostructures like this. And finally, with the help, we can collect those nanocrystals or nanoparticles for further experiments. Digital microfluidics, here they use this chip for chemical reaction. Of course, in this case, a multiple reaction. So just using a single chip, 4 to 5 reactions can be performed simultaneously. So finally, the researchers got product like this, and this product was used for analysis like mass spectrometry and NMR analysis. In our group, now we are making these digital microfluidic devices for some chemical reactions and also for surface enhanced Raman spectroscopy. Here you can see a tiny droplet is moving from one slide to another slide. So this is just a normal water, but we are planning to make some chemical reactions using this microfluidic chip, and Raman spectroscopy will be used for monitoring the reaction. So reagent A is coming from this side and another reagent is coming from this side. We can mix it and we can wait for the product formation. So why do we, so far we saw like why we need microfluidic devices for synthetic chemistry. But now let's see, why do we need analytical techniques for these microfluidic devices? So this is what I mentioned. So if you are making a chemical or biological reaction, then we need to know what is there, like what is the product or what is the intermediate. For that, we need analytical techniques. So one thing, if we got mixtures or if we got a biological sample like urine or blood, basically we will have matrix effects. So sometimes we cannot get the exact signal from our target molecule. So it's always better to do pre-treatment or in some cases separation like HPLC to separate the multiple components, and we can detect each component using different techniques. So we need some separation techniques for separating a complex mixture. Then optical detection methods like Raman spectroscopy, fluorescence, or IR. Since we are using glass slides, so we will not have any limitation or problem with this online monitoring. We can just hit the slide with an optical microscope like this. We can use SIRS or fluorescence for monitoring. Then, of course, for mass spectrometry, we cannot do the experiment, the detection inside the channel. For that, we have to spray it. So here it's electrospray ionization. It's a method to spray the target molecules into a mass spectrometer. So now let's see the application or the relationship between microfluidic chip and chromatography. So this is from our group. In this work, we combined droplet microfluidics with chip chromatography. So here we got inlet for mobile phase for separation, and we got the packing channel here. So packing channel means we have to use this channel to pack the column. Like we have to make our own separation column inside this channel, let's say 1 centimeter. So by using this channel, we can pack this channel with separation column, then inlet oil phase, that's for making droplets like this. So we injected a mixture of analytes, and we used this column for separation. And finally, we detected those mixture like different components using fluorescence technique. So this is one application. That means we can separate the mixture and we can clearly see the signals from each component or each chemical. Chip chromatography and mass spectrometry. So in the previous slide, we used fluorescence to detect the analyte, but in this case, it's for a flow reaction. So this is the reaction, and we use this channel. So I said we got multiple turns like this to manipulate or to mix the fluids more efficiently, and we got a reactor. This is reactor, and we got a column, HPLC column inside the same chip. So the products were separated, and finally we sprayed that analyte into a mass spectrometer, and we got the signal. So one option, you can continuously flow the reagents and measure, and also you can perform like stop flow. You can stop the flow, carry out the reaction, then finally we can spray and detect the products. Here, heterogeneous catalysis with chip chromatography and mass spectrometry. So in this, one step ahead, like we used a catalyst and we performed a chemical reaction. So it's a nanoliter sized reactor. We got this amount of catalyst. If you're doing some normal traditional reaction, maybe we may need this amount of catalyst. But when we are using microfluidic devices, we just used the micrograms of catalyst, and that catalyst can be reused. So that is another advantage. And with this, we performed a chemical reaction and detected using mass spectrometer. Here, mass spectrometry for the investigation of continuous flow reactions in microfluidic chips. So we used a glass chip and a flow reaction, continuous flow reaction. This is the one. And here we used IR laser to spray the analyte. So previously I mentioned easy spray, electrospray ionization. That is one method to spray analyte into mass spectrometer. But here we used a laser, infrared laser, and that can evaporate and spray the analyte into the mass spectrometer. So we can adjust the flow rate. So the flow rate can affect the reaction rate. And here you can see the monitoring. Continuously we can measure using mass spectrometer, and we got signals from the product. And in this case, microfluidic chip with the fluorescence technique. Again, a chemical reaction. So these are all inlets for the reagents, and we got different products like R and S, salicylanol, but we used fluorescence here. So in this case, we don't need to spray or we don't need to wait for the outlet thing. Here, we just did the real-time monitoring. So we can continuously measure this part. So here we can see the single photon counting, let's say lifetime measurement. So by using this lifetime data, we can find out whether the product is R or S, or the reaction is complete or incomplete. So we can use fluorescence like optical methods for reaction monitoring. So it's an emerging field like droplet sorting, or you can say single cell analysis. So in this case, we used electrodes and we use fluorescence lifetime measurements for droplet sorting. So we got a mixture of droplets, let's say 2 different dyes, dye A and dye B. Now we need to sort it. So dye A should go to the left side and dye B should go to the right side. So based on this fluorescent data, lifetime data, we can program the electrodes. So the electrodes will be activated. So if the detector says A, then that will go to the right side, then another signal will go to the left side. So this kind of experiments will be useful for some biological analysis. Let's say cancer or non-cancer cells or fatty, non-fatty cells. So if you want to separate two different cells, then we can use this kind of sorting method. So one will go to the collection reservoir and the other side, maybe we can direct it to a vase. Then we can collect the really good cells for further experiments. In this case, infrared spectroscopy with microfluidics. So in our case, like we did infrared spectroscopy measurements using pellets. So we need some sample and we make pellet, and finally we can place it inside an infrared spectrometer. But in this case, we can just do the infrared spectroscopy measurements inside the chip. But of course, we need a window material like zinc selenide or germanium crystal, something like that. But here they used a zinc optical window to transmit or to allow the infrared radiation through these channels. And continuously they measured the spectra. And with this data, we can tell whether the reaction is finished or whether the product is formed or not. So that is one way. We can incorporate IR also, this microfluidic devices. Here, spectroelectrochemistry with microfluidics. I said electrodes can be integrated inside a chip. Here you can see, and a microfluidic cartridge with a portable instrument where we can do electrochemical measurements. And in some cases, spectroelectrochemistry, that means we can perform both spectroscopy measurements and electrochemical measurements. The combination of spectroscopy and electrochemistry is called spectroelectrochemistry. So we can perform this with a microfluidic device. Even without a microfluidic device, spectroelectrochemistry is another field. Recently, we published this work. We combined chip chromatography with ion mobility spectrometry, IMS. So in this chip, in previous slides, we saw we sprayed the analyte to a mass spectrometer, but here it's a portable ion mobility spectrometer. And here you're seeing a microfluidic chip, and these are all fluidic connections, screws to just tighten it. So the samples can be injected through these channels and we can separate it using a chip. And finally, we sprayed it to IMS, and we published this recently in Analytical Chemistry. Now let's look into another field, microfluidic SIRS. That means microfluidic surface enhanced Raman spectroscopy. This technique carries our Indian scientist, Dr. Sir C.V. Raman, who discovered this technique in 1928. So that's why Raman spectroscopy carries our Indian name. So basically it's about Raman scattering. So it's inelastic scattering. This technique can give fingerprint information about different chemical and biological molecules. So nowadays researchers are using SIRS for ultra-sensitive detection. But in our group, we combine this particular technique SIRS with microfluidic devices because of some advantages. But first, let's see what is the difference between Raman spectroscopy and surface enhanced Raman spectroscopy. Raman spectroscopy, in this case, we don't use nanoparticles. That means we just use our analyte like this, and we can excite this with laser. But usually the problem is Raman scattering process is a weak process, so the technique itself suffers with sensitivity. We cannot detect ultra low concentration of molecules using Raman spectroscopy. Let's say if we have micromolar or nanomolar concentration of molecules, then it is really difficult with Raman spectroscopic technique. In that case, we have to go for SIRS. So this technique was discovered in 1974 by Fleshman and his colleagues in the UK. They accidentally discovered this technique. So they found out that nanoparticles can be used for signal amplification. Actually, they did not explain it in the first paper, but later from the US, Professor Richard, he went and he explained the reason behind this enhancement. So there are two mechanisms. First thing, electromagnetic enhancement and chemical enhancement, but that's too much now. So let's just see nanoparticles, they are acting as nano amplifiers. So we can detect even ultra low concentrations like nanomolar or even femtomolar concentration can be detected using SIRS. In some cases, even single molecule can be detected. Advantages, why we have to combine these two different techniques like microfluidics and SIRS. So with microfluidics, we can integrate multiple functionalities. That means we can perform SIRS and also fluorescence, and finally mass spectrometry is also possible. So we can perform 3 different analytical characterization using a microfluidic device. The next thing, it's reproducible, automation is possible, and portable. We can also make nanomaterials using microfluidics. So we can use that for SIRS. On the other side, SIRS can give fingerprint information about a wide variety of chemicals and biomolecules. That's a good thing. Then it is sensitive and selective. We can do multiplex detection also. And another big advantage is non-destructive. So we can do real-time monitoring. But with, for example, with mass spectrometry, it's destructive. We cannot go for further measurements. But with Raman and fluorescence, they are, especially with SIRS, they are non-destructive. And yeah, of course, real-time analysis is possible. And when compared to IR spectroscopy, Raman is suitable for aqueous samples also. No water interference. Water is a weak Raman scatterer. So we can detect biological molecules and also aqueous samples using SIRS. So how the setup will be. For microfluidic SIRS, we need a microscope, Raman microscope. Here you are seeing a Raman microscope with laser. Then this is the sample holder where we place our chip like this. And the laser will be excited from this part, and here it will be scattered. Then we have to collect that light, and by using this detector, usually CCD, we can analyze the data and we can get the data from the computer. A typical picture showing the same setup, microfluidic chip, and the scattered light will be collected and it will be analyzed using a spectrometer. So we use this microfluidic device for reaction monitoring, again, on-chip monitoring of chemical synthesis. So in this case, it's a droplet microfluidic device where we performed a chemical reaction and continuously we measured the data, Raman data. We got Raman data, and by assigning the particular peak, let's say this peak is from the product, then we have to monitor that particular peak and we can make a curve like this. So when the reaction is started and when it is completed, we can get the data from the Raman spectrum. In this case, microfluidic SIRS for foodborne pathogen detection. So this is from a different group where they mixed analyte and like foodborne pathogen and nanoparticles from this channel, and they concentrated this nanoparticles into the center part, and they were able to detect different foodborne pathogens using a microfluidic device. So this kind of work can be used for food analysis, for food science. This is from our group, application in food science. In this work, we detected melamine in milk samples. So melamine is a toxic chemical. It's a food adulterant. It should not be mixed with milk, but there were some accidents. So it is important to detect these kind of adulterants in food samples. So in this case, we fill the reservoir with the sample containing melamine, milk sample containing melamine, and we applied a high voltage. Then with the help of electrokinetic flow, that means we can selectively transport the molecules from one side to the other side. So we place the nanoparticles coated membrane in the middle, let's say here. So top channel and bottom channel. The top channel was filled with melamine and milk. We selectively transported the melamine molecules from the top channel to the bottom channel. So when they cross this nanoparticles, we can detect using laser. So we were able to detect the melamine peak in milk samples. So spiked milk, if you detect just the real sample, like milk sample, we will not get the melamine signal because matrix effect. So we have to separate our target molecule from the matrix. But after that electrokinetic transport, we were able to see the particular melamine peak. This is from pure melamine, and now we transported that melamine from spiked milk. You can clearly see the peak. So this is one application of microfluidic source in food science. This picture just shows the morphology of that membrane, a bare polycarbonate membrane, and we coated that membrane with nanoparticles. Nanoparticles, they are nanoamplifiers. They just enhancing the molecules, melamine molecule signals. Microfluidic technologies for environmental applications we can use for different analysis, let's say the class of pollutants like sulfite, nitrate, dyes like methylene blue, malachite green, and heavy metal ions in different food and also for environmental samples. And here you can see different techniques can be used, optical techniques like fluorescence, Raman, IR, electrochemical measurements are also possible. Spectrometry can be used. So this kind of applications are also possible using microfluidic technologies. Now let's move on to the next topic, maybe the final topic, organ on a chip. So now we can make a single organ inside the chip. So why do we need this kind of devices? There are different advantages we will see now. So here you are seeing different organs like liver, heart, brain, lung, kidney inside a chip. Same microfluidic chip, that means a microscope slide. We have to bond it and we can control the parameters very precisely. So advantages and disadvantages. So the main advantage of organ on a chip or microfluidic chip is fine control over microenvironment, good mass transport, and ability to integrate various sensors. So that is one good thing. We can see one application in this particular field. Here, they mimicked used a bio-inspired microfluidic device by integrating a porous membrane for biomolecule cleaning. A membrane was used to blood, simulated blood, and dialysis can be performed using this kind of devices. Maybe in future, the vision is like this. If you have a device like this, then you can blood field. So there are different companies, they are making these microfluidic devices for both commercial applications and also for some scientific applications. Some laboratories, even in our group, we buy some complicated microfluidic devices from companies. So these are all the top leading players in microfluidics device market. And in the future also, the demand will increase. It seems like the top companies like Abbott, Siemens, Agilent, Micronet, and some companies in India like Achira Labs, they are making point of care diagnostics, POC devices, and microfluidic devices for biomedical applications too. So for scientific experiments, I would say it's better to prepare our own chip But for some cases, if you want to buy a chip, then a single microfluidic chip may cost around $60 to $100. If you want to know more about the products and the companies working on this particular field, you can check this article, Nature Methods. They collected all these details and they presented nicely. These are all the companies, they are making microfluidic chips. And of course, they are bringing out some new applications. Conclusions. Microfluidic devices can be used for different fields, especially for chemical synthesis and even nanomaterial synthesis. In the biological part, we can also use it for single molecule analysis, organ on a chip, single cell analysis. And another interesting direction is hyphenation with multiple analytical techniques. Same time, at the time, we can perform 3 or 4 analytical experiments, like using different techniques. Decreases sample and reagent consumption. Cost-wise, it's good and shortens the time of experiment. It's reproducible and reliable. So take-home message for both students and faculty, also for researchers. So it's an interdisciplinary field. It deals with physics, chemistry, biology, and engineering, promising applications in both chemistry and medicine. So if you are interested in this field, if you want to do research in this particular field, you can think about any one of these directions. So first, rational designs for the fabrication of microfluidic devices. Companies are working on this direction. They are making new complicated devices, rational design. So if you are running a research group, you can just focus on making new microfluidic chips, but you need the infrastructure. If you are interested in theoretical part, if you are an engineer with good computational skills, then you can think about machine learning and artificial intelligence for data analysis. So I said we are using multiple techniques, different analytical techniques. So we get usually, in some cases for biological samples, we get more complex data from the system. So it is difficult to analyze the data manually, visually. So in that case, machine learning and artificial intelligence can help. There are some groups in Canada and the US, they use machine learning and artificial intelligence for data analysis. Let's say if you get a Raman spectra, SIR spectra with multiple peaks, then it is good to use any computational methods to analyze the data. Next direction, novel applications in synthetic analytical chemistry or biology. You can prepare your chip, or you can get chips from your collaborators or companies. Then the role will be bringing out some new application using that device. In food science or environmental science, that's fine, but a new application. Then another direction, hyphenation with the different analytical techniques. We are also working on that direction. So we can couple different techniques, let's say mass spectrometry and SIRS, fluorescence and SIRS, fluorescence and IER. You can couple 2 or 3 techniques using this microfluidic device, and let's try to get as much information as possible. So we can get more information about the target system. That is another direction. So with this, I would like to thank my lab members. This is our research group in Leipzig. And I would like to thank the German Research Foundation, DFG, for the funding and support. So thank you for your attention. I'm glad to answer your questions now.
If you have any queries, you can pose the question. You can put up the question in chat box, or you can feel free to interact with the resource person.
Hi, my name is Rohan, and I thank you for a very good, wonderful presentation. I actually had a list of questions, so perhaps I will email you separately, just in the interest that
Oh,
everybody gets a chance to ask you questions. But my most pressing question is, what is the limitation? Like you've given the example of creating masks, right? The mask can be created as simply as using a laser printer. Is that correct?
laser printer is one option.
Okay. What about 3D printing?
3D printing, that's an emerging field.
Yeah,
Actually,
but
we
what
got
would
a
be the
new print.
limitations with that?
Limitation, first thing, we have to practice more. Um, we have to, how to say, we have to make the new design by ourselves. So it's not already published, or we cannot get the recipe from somewhere. So we have to make it. So that takes time. And even now, like for the past 3 or 4 months, we are just training ourselves to use the printer, how to use it. That's the big limitation now. Yeah.
Okay, but not in terms of like channel width and everything, because even that has a limitation, correct?
Channel limitation is not a big problem.
Okay. So for example, something like the photoresist, can it be 3D printed into very small, let's say 100 micron height or diameter channels?
But so far it's not necessary. Sometimes, like, why do you want to make it you want to make it more smaller or bigger?
Yeah, make it more small.
Why do you want to do that?
I'm just curious whether it's possible or not.
Okay, yeah, in some cases it's possible, but it's not good to have really small channel. So that is one option, but 3D printing is an emerging and interesting thing. We can use it. There are some advantages, of course.
Okay, cool. Like I said, just in the interest that everybody gets a chance to ask other questions, if you could share your email, or if I could perhaps get an email address where I can contact you, then maybe we can discuss this a bit more further, if that's okay with you.
Yeah, sure, I can share my email ID. Yeah.
Okay, thank you very much.
Welcome.
Sir, hello, sir.
Yeah.
Hello, sir. Yes, sir.
Hello.
Sir, for urine contamination, for urinary tract infections,
Mhm.
to detect the bacteria, especially if I design by using silver nanowire microfluid, sir, is it possible? Only by I have only FE, SEM, XRD, and XPS like instruments. Can I design with this?
I think it's difficult. You are talking about the imaging techniques, but it is difficult to detect a pathogen or bacteria by just seeing an image. So it's better to perform some measurements, like even SIRS is also possible, Raman spectroscopy. You have to get the fingerprint information about the bacteria or microorganism.
Sir, Raman spectroscopy may be possible.
Yes, possible.
Sir, what type of application in this case can you suggest?
Sorry, what type of application in?
Raman, by using the Raman spectroscopy, exactly what I can detect.
Okay, it's a big question. So I can say Raman spectroscopy can be used for detecting food contaminants, like some chemicals. That is one part. Another thing, you can also detect bacteria, virus, some other microorganisms using Raman. Then in energy field also, you can monitor a chemical reaction, or you can monitor electrochemical reaction using Raman spectroscopy.
I am preparing also electro catalyst for
Mhm.
fuel cell, but I want to
Okay.
just changing towards this directions by listening your topic.
You want to, sorry, it's not clear.
exactly I am preparing electrocatalyst for the fuel cells, alkaline fuel cells I am using. But by listening your talk, I am very inspired now to work.
There are some groups actually in China. They use Raman spectroscopy for energy devices. So that's an interesting and more famous topic. You can use Raman for electrochemical measurements.
Yes, sir. Thank you, sir. I will later contact you by your email.
Okay, fine.
Thank you.
Hello, sir.
Yes.
Sir, difference between fused deposition modeling and selective laser modeling.
Selective modeling and what is the other thing?
Fused deposition modeling and selective laser modeling. Major difference, sir.
I cannot answer this question simply, but because I don't get what kind of difference you're expecting, like.
So any major difference, sir?
So it's a different method. So in cases of application or
application also.
No, the modeling thing is just for how to say, just for making channels and making device. Application will be different part. It's a different part. So if you are specific about the application, then maybe we can talk about the channels or modeling thing. If you're not sure about the application, then it's difficult.
Any other questions? Any other questions from participant side? Sir, this is one question from Tyagarajan Meera. Which type of nanoparticles can be used for amplification process?
Okay, so plasmonic nanomaterials, first thing, gold, silver, and copper. These three materials are more common. So if you are interested in such measurements, then I would say you can start working with gold and silver nanoparticles.
Thank you, sir. This is another question from Dr. Sri Shakti Priyadarshini. Can we examine the gaseous samples, for example, analyzing pollutants in air? I think you have shown in the slides.
Yes, it is possible. You can use IR or Raman spectroscopy for measuring gaseous sample. That is possible.
Another question, sir. Any reaction depends on its temperature and pressure in case. In that case, how that is controlled in those chips? Whether it depends upon temperature or pressure.
Temperature, of course, we can use really tiny hot plates to increase the temperature of the chip. We can control the temperature. For pressure, usually you can control it by the syringe pump. So what's the flow rate? The flow rate depends on the pressure.
Yes, I think you have answered Mr. Anil Raj question also. It was also about high pressure and high temperature reaction. Can such devices sustain? I think you have answered it. Sir.
Yes, yeah.
Participants, any other question? Any other question to ask with resource person? So really, it is an interesting webinar, sir. I'm sure we have, Indian researchers explore to new technology. Microfluidic, it is my opinion. as till now we are adapted to the conventional research in chemistry, especially wet lab like those. So hope your presentation will bring a new revolution in the field of analytical chemistry. At the same time, your presentation gave a new idea pitch for start up the new projects or new research. Research. At the same time, it is very great to hear that this microfluidics plays a vital role in pollutant detection, cancer detection, at the same time in dialysis field too. Really, we just wonder how this chip system plays a role in cancer detection and all. Thank you so much, sir. Thanks for accepting our invitation and give such a wonderful session to the participants. Let me call upon Dr. Deepa to present the vote of thanks. Deepa, ma'am. Deepa, ma'am, are you there?
Yeah, I'm there. Okay. Before proceeding with my vote of thanks, another important announcement. There is another interactive webinar scheduled tomorrow at the same time between 3:30 and 4:30 p.m. Let me propose vote of thanks. Respected resource person of the day, Dr. J.R. Rajapandian, research scientist, Institute of Analytical Chemistry, University of Leipzig, Germany. Most respected CEO and Secretary of NGI, Dr. Krishna Kumar, beloved principal of NIET, Dr. Maniarasan. I thank them in their absence here. HOD of Science and Humanities, Dr. S. Shalini Bhakyam Kamala, HOD from various streams of engineering, Administrative Officer of NIET, Mr. Sudarmani, media team of NGI, Mr. Roshan and his team, academicians from different countries, participants, I express my gratitude for your presence to make the webinar a successful one. Once again, my special gratitude to our resource person for making the session an informative one. Thank you all. Join us tomorrow.
Shalini, ma'am.
Tell me, Dr. Vrinda. Yes, yes. Let us wind up the session today and we'll meet tomorrow.
Thank you, sir. Thank you once again for make the session interesting. Of course, it is we are learning new things even yesterday and today also. We are exposing to new many ideas and new technologies. Thank you, sir. Thank you so much.
Welcome.
Actually, we all thought it is a chip is related to lab related. We wonder how it. Myself and Brinda, we discussed how it would happen because the only course what we left out during lockdown is laboratory course. But by the title itself, you attracted us. The session is so informative and we listened carefully. It was useful, sir. Thank you.
Thank you. Welcome. So, bye. Yeah.
Thank you, sir. I think this one session is not enough to get a full knowledge on this microfluidics. Of course, even today I have searched the microfluidics in Google and I gathered few things about it only today. So after your session, it was so interesting.
Okay, fine.
Thank you, sir. Thank you so much.
Okay, then. Yeah, bye.
Bye, sir. Have a nice day.
Bye. Yeah, you too.
Thank you, sir.
Thank you, participants. You can take a leave and let's meet tomorrow.
Thank you. Thank you, participants.
Sudar sir, Sudarmani sir, Roshan sir, we can close the session.
Yes, ma'am. Hello, ma'am. Ma'am, we request for the Gmail address for Rajapandian, sir.
Okay. Brinda, can you please share it?
Yes, I have given in the chat box, ma'am. Ma'am, you can scroll down the chat box and you can see. Or else let me type now.
Okay, ma'am. Okay.
Type again. So that many people ask for it and you collect his PPT too and then let's share through their mails.
Yes, ma'am.
Okay,
For
ma'am.
sure, ma'am. Ma'am, Shalima may have shared the mail ID of resource person. Participant, please note down if you want to interact further.
Thank you, ma'am. Thank you.
And let us send it through a mail also, Brinda. That will be useful to the participants, those who left already. So
Yes,
let's
yes.
share the PPTs as well as mail IDs of our all 3 days resource person, right?
Yes, ma'am. Sure, ma'am. Sure, ma'am.
Okay. Thank you, Brinda. Thank you, Dr. Deepa, and thank you all participants.
Thank you, ma'am.
Sudhir, sir. Sudhir, sir.
ஹலோ மேடம்.
Sir.
ஹலோ.
Yes, sir.
இல்ல ரெண்டு ஃபீட்பேக் லிங்க் இருக்கு, அது எது கரெக்ட் மேடம்?
Sir, இப்போ நான் ஒன்னு போஸ்ட் பண்ணிருக்கேன்ல சார்.
ரமேஷ் கேகேஆர் அப்படின்னு ஒருத்தர் பண்ணிருக்காரு. அப்புறம் மேடம் உங்க பேர்ல ஒன்னு இருக்கு.
Sir, you can go with my name, sir. என் பேருக்கு கீழ ஒரு ஃபீட்பேக் லிங்க் போட்டிருக்கேன், அது ஃபில் பண்ணிடுங்க.
சரி சரி மேடம், சரி.
ஓகே சார், ஓகே சார். மேபி ஷார்ட் ஃபார்ம் எங்க டீம்ல இருந்தா ஷார்ட் ஃபார்ம் போட்டிருப்பாங்க சார்.
சரி சார். சரி மேடம். அப்ப அதான் ரெண்டுமே சரி ஒண்ணுதானே?
ஆமா சார், ஒண்ணுதான். இப்போ என் நேம்ல போட்டது நீங்க ஃபில் பண்ணுங்க, நோ ப்ராப்ளம்.
சரி, நீங்க உங்க பேர்ல இருக்க ஃபில் பண்ணிடுறேன்.
எஸ் சார், ஓகே சார்.
சரி மேடம், சரி மேடம்.
அகைன் நான் இப்போ ரீபோஸ்ட் பண்ணிருக்கேன் சார்.
சரி மேடம், சரி மேடம். தேங்க்யூ மேடம்.
தேங்க்யூ சார், தேங்க்யூ.
தேங்க்யூ மேடம்.